Welcome to Tomorrow Unveiled, the show that takes a comprehensive look at the complex science shaping our health, vitality, and future.
Today, we are witnessing, it's really a fundamental shift in how we approach the human body.
Yeah.
For decades, the entire conversation around longevity was just about extending lifespan.
Right, adding more years, but not necessarily better years, often years of decline.
Exactly. The new scientific paradigm is completely different. It's all about functional life extension, adding years of robust, active vitality.
That distinction vitality over just survival, it feels like the most critical narrative in modern biology right now. And what makes this look into the research so exciting is seeing how three previously separate fields are now converging.
Yeah.
We're not talking about just one magic pill anymore. It's a full stack approach, blending this high-tech engineering with the basics, foundational biological maintenance.
Absolutely. The stack of sources and intelligence our research team compiled highlights this powerful convergence across three main pillars. First, you have programmable immunotherapy, where we are literally engineering the body's own repair systems.
Okay.
Second, metabolic foundations. This is what governs the absolute, the rate-limiting steps of aging, the basics we so often overlook.
And the third.
The third is algorithmic biology. This is where artificial intelligence comes in to find the subtle patterns and novel targets that, frankly, human intuition just misses. Our mission on Tomorrow Unveiled is to unpack the data points that are moving these intersecting fields from theory into clinical reality.
And hopefully provide some genuine aha moments for you, the learner.
Yeah.
Okay, let's unpack that first pillar because it really feels like we've crossed some kind of threshold in medicine. We're moving past just supporting the body and into actively engineering its repair.
We are.
And the biggest news here seems to involve repurposing a really powerful cancer treatment, CAR T cells, the so-called living drug, and deploying it against the root causes of aging.
It's truly transformative. The core breakthrough that our research team highlighted involves using these engineered T cells to target senescent cells.
The zombie cells.
The zombie cells, exactly. For anyone new to the term, these are cells that have stopped dividing because of damage, but instead of dying off like they should, they just linger.
And they're not just passive, right? They're actively causing problems.
Oh, absolutely. They secrete this nasty cocktail of inflammatory compounds. It's called the SASP, or senescence-associated secretory phenotype. And this toxic stew is a major, major driver of the chronic inflammation and tissue dysfunction that we associate with getting older.
And historically, getting rid of those zombie cells has been, really difficult because they've evolved these defense mechanisms.
They use specific proteins like BCL-XL and BCL2 to basically evade the body's natural cleanup crews. But CRT cells are the ultimate in precision medicine.
So how do you program them? How do you tell a T cell to find only an aged senescent cell and not touch a healthy, functioning one right next to it?
That specificity is the whole game. It's the key challenge and the key breakthrough. To do this, you need a unique surface marker, something that only appears on those aged dysfunctional cells.
Like a little flag they wave that says, I'm a problem.
Precisely. And the research we reviewed identified the urokinase plasminogen activator receptor, or UPR, as that unique marker.
UPAR. Okay, so UPI is the beacon.
Exactly. The engineered CAR T cells are then equipped with a receptor that recognizes and binds only to this UPR protein. They become these customized guided missiles designed specifically to locate and eliminate those zombie cells. It's a level of targeting it's far more precise than traditional drugs.
That makes sense. So once these customized T cells were put into aged animal models, what did the results actually show? Beyond just killing off some zombie cells, what was the functional outcome?
The results were genuinely shocking, especially in the gut. The T cells actively patrolled the tissues. They located the UPR-positive senescent cells and eliminated them. But the functional outcome went so far beyond just cleaning house.
What had happened?
We saw a significant restoration of the gut's epithelial barrier. It actually reversed the common leaky gut phenotype that's associated with inflammation and aging.
Wow. Just restoring the gut barrier alone is a huge win for systemic health. So much chronic disease is tied back to gut inflammation.
It is. But the research team also highlighted something even more profound about regeneration. This is where it moves from simple damage control to active reversal.
Okay, I'm listening.
The team found that removing the senescent cells actually restored stem cell function inside the intestinal crypts to levels that were comparable to young animals.
Whoa. Okay, so that's a massive insight. It implies that these zombie cells aren't just sending out inflammatory signals.
No, they're not.
They're actively putting the brakes on the body's own built-in regenerative potential.
Precisely. Think of the senescent cells as a biological system jam. They hijack the local environment and they inhibit the stem cells. So when you remove them, you release that regenerative brake. The intestinal crypts, the nurseries for new gut-lining cells, can regenerate with the vigor of youthful tissue.
It suggests a potential for systemic rejuvenation that just wasn't possible before. What about durability? If this is a living drug, a single dose has to last, right?
The durability is a huge piece of this breakthrough. The studies show that a single administration of these anti-UPAR CAR T cells provided benefits lasting for at least 1 year in mice.
Which is a huge fraction of their lifespan.
A very significant fraction. The CAR T cells persist in the body, providing this ongoing surveillance, preventing the zombie cells from building up again.
But I have to ask, if this is a living drug designed to persist for a long time, doesn't that raise safety concerns? I mean, if these cells are active for years in a human, what's the risk of some off-target effect emerging 5 or 10 years down the road?
That is the main concern, and we have to acknowledge it with any persistent gene or cellular therapy. While the specificity for UPR is high, the long-term immunological safety profile that remains the primary focus of ongoing research.
So it's a trade-off between power and control.
It is. The engineering has to be refined to ensure that even if the T cells persist, they remain tightly regulated. The field is still learning how to achieve that perfect level of control.
A necessary caveat. Okay, let's pivot slightly to the parallel path. While CRT cells are this living therapy, there are also huge advancements in small molecule senolytics. We know the first generation had issues with selectivity, right? Things like platelet damage.
They did. That first generation, while a revolutionary concept, had unwanted side effects because the targets they hit were also present on healthy cells. The intelligence we reviewed shows a clear push toward dramatically improved safety through molecular precision.
So?
We're now seeing the development of new targeted senolytic candidates. They selectively degrade the very proteins, specifically BCL-XL and BCL2, that the senescent cells use to cheat death.
So these new drugs are basically stripping the zombie cells of their armor, but leaving the healthy cells nearby untouched?
Exactly. It's precision pharmacy. The studies confirmed that this highly targeted approach successfully removed senescent liver cells, which is critical for treating conditions like liver fibrosis, while crucially avoiding those previous toxic side effects.
And that improved safety profile is what's letting them move into human trials faster.
It is. And we're already seeing this enhanced safety translating into clinical synergy. The combination of these new senolytics with other treatments, that seems to be where the field is really heading.
Combination therapies.
Yes, they're emerging as a profound strategy, particularly in cancer. We reviewed data from trials where specific senolytics are being combined with anti-PD-1 immunotherapy. The idea is that clearing out the cellular junk enhances the immune system's ability to fight the cancer.
And the results of that combo.
They're compelling. In a phase 2 trial, this combination achieved a significant clinical response rate while dramatically lowering the severe adverse effects you'd see with conventional chemotherapy. It really validates the idea that aging therapies, they're not just for aging. They can make other medicines work better.
Okay, moving from the immune system's programmable soldiers to a more foundational regulator. Let's talk about the Klotho factor. This protein has been called a master regulator of aging.
And for good reason. Its regulatory scope is vast. Klotho levels decline significantly as we age, and this decline is implicated in everything from kidney disease and cardiovascular aging to cognitive decline.
So the research our team analyzed looked at gene therapy to restore Klotho levels.
Yes, and the results showed that restoring Klotho is the epitome of what we're calling functional life extension, not just adding years.
What were the numbers?
We saw an extension of lifespan in mice by a substantial 15 to 20%, which is significant.
Yeah.
But the truly remarkable part was the breadth of functional gains.
Like what?
The treated mice demonstrated enhanced muscle strength and grip endurance, significant improvements in bone density, and most importantly for us, profound cognitive performance improvements.
So they were stronger, their bones were healthier, and they were smarter.
They showed superior memory and learning ability. It suggests this therapy was actually driving neurogenesis in the hippocampus. A single gene therapy intervention simultaneously addressing strength, structure, and the brain. It's a multi-system fix.
It's like Clotho is an overarching systemic signal, pushing the whole system back towards a more youthful state.
That's a great way to put it. It coordinates repair across multiple tissues. And this ability to restore a master regulator or to program immune cells to clear debris, it powerfully demonstrates this shift toward actively reversing the molecular drivers of aging.
If aging is a complex systems failure, then Clotho is like rebooting the whole operating system, while CRT cells are running specialized defrag software.
A perfect analogy.
But this high-tech engineering only works if the hardware itself is stable, which brings us to our next pillar.
And that's the critical link. As incredible as engineered T cells are, they can't work miracles if the body's foundation is crumbling. This is where high-tech intervention meets the most essential rate-limiting factors. And the intelligence we gathered had some truly eye-opening data here.
You're talking about the data on sleep, right? Which kind of challenges the old diet and exercise first mantra. For years, those were the undisputed top two.
They were, but a massive analysis, looking at millions of data points, established a stunning statistical link. Researchers found that insufficient sleep, defined here as less than 7 hours a night, was a stronger predictor of decreased life expectancy than both diet quality or physical inactivity.
That is a huge shift in perspective. So prioritizing your sleep is actually more critical for survival than hitting the gym?
According to this data, yes. It ranked second only to smoking in terms of mortality risk.
We need to understand the mechanism there. Why is sleep suddenly so critical?
Because sleep isn't just rest. It's the mandatory maintenance cycle for physiological repair, especially in the brain. The primary mechanism is the glymphatic clearance system.
The brain's plumbing and waste removal crew.
That's it. During the day, our brain cells are densely packed. But when we enter deep, slow-wave sleep, the glial cells, the supporting cells around the neurons, they actually shrink by about 60%.
60%?
Yes. This shrinkage dramatically increases the space between the cells, allowing cerebrospinal fluid to rush in and effectively wash away all the metabolic waste that built up during the day.
So the brain literally opens up the plumbing channels during deep sleep.
Precisely. And it's during this flush that neurotoxins like beta amyloid and tau, the proteins implicated in Alzheimer's, are clear out. If you chronically deprive yourself of sleep, you are severely impairing this clearance process.
You're essentially running a chronic toxic buildup operation in your own brain.
Exactly. And that buildup directly translates into the cognitive and systemic decline that feeds those mortality statistics. The research also highlighted the cardiovascular risk. Untreated sleep apnea, for instance, forces the body into chronic overdrive.
Right.
And that's directly linked to accelerated heart aging and arterial stiffening. The data is unequivocal. For any advanced longevity strategy to work, 7 to 9 hours of quality sleep is a non-negotiable prerequisite.
This foundational theme continues when we look at the shocking link between obesity and accelerated neurodegeneration.
It's a stark connection. A recent study tracked participants for 5 years and found that obesity acts as a direct molecular accelerant for Alzheimer's. The study showed obesity speeds up the increase of specific Alzheimer's blood biomarkers, like tau-217, by up to 95% faster compared to healthy weight individuals.
95% faster. That is a massive signal. What's the mechanism? What's the bridge between fat tissue and tau tangles in the brain?
It's what the researchers call the inflammatory bridge. Visceral fat, the fat around your organs, is highly active. It secretes potent pro-inflammatory cytokines into your bloodstream.
And those cross the blood-brain barrier.
They do. They trigger neural inflammation inside the brain itself, and this inflammation impairs the machinery that's supposed to be clearing out those toxic proteins.
So excess weight creates this constant state of low-grade systemic alarm that directly impedes the brain's cleanup crew.
That's half the battle. The other is insulin resistance. There's a crucial enzyme called insulin-degrading enzyme, or IDE. Its job is to relieve insulin, but it also removes amyloid proteins. When you have chronic insulin resistance, that enzyme is so busy clearing out high levels of insulin that it creates a traffic jam, slowing down the removal of amyloid.
So it's a compound failure. Clearance failure from bad sleep and chronic inflammation from metabolic overload.
The ultimate 1-2 punch. And this leads us to a really crucial diagnostic caveat that the research team identified. As we start relying more on these blood biomarkers like p-tau-217 for early Alzheimer's diagnosis, we have to be careful.
What's the trap?
Other studies have shown that reduced kidney function, which is really common in aging, can artificially inflate these Alzheimer's biomarker levels.
How?
The kidneys are responsible for filtering these proteins out of the blood. If they aren't working well, the proteins accumulate, which could lead to a false positive or an overestimation of the brain pathology.
So a doctor might see a high p-tau217 and think high Alzheimer's risk, when the real issue is declining kidney health.
Exactly. Future diagnostics have to account for renal health, for EGFR, to accurately assess brain risk. It demands a holistic view.
Okay, let's unpack this. The idea that something as simple as sleep or maintaining a healthy weight acts as a fundamental, non-negotiable prerequisite for high-tech longevity interventions is a huge insight. If you're finding this insightful, please take a moment to like this video, subscribe to Tomorrow Unveiled, and consider hitting that new hype button to help us reach more viewers. And of course, be sure to turn on notifications so you never miss an update on the future. Now, let's dive even deeper, right into the cellular power grid, the mitochondria. Their efficiency is everything, and new discoveries are showing they're far more organized than we thought.
The old analogy of the mitochondria as a single powerhouse is just too simple. It's more like a complex, interconnected power station. For years, we thought the components of the electron transport chain were just floating around. The super complex hypothesis completely overturns this.
And what does that hypothesis state?
It states that these components don't flow freely. They cluster together into these stable, highly organized structures called supercomplexes or rastrosomes.
Why does that clustering matter so much for longevity?
Because organization dictates efficiency. Think of an assembly line. If the stations are close, the product moves quickly with less waste. When the ETC complexes cluster tightly, ATP production is far more robust, and, crucially, it significantly reduces reactive oxygen species, or ROS.
The damaging cellular leaks.
Exactly. ROS are the metabolic exhaust fumes that cause DNA damage and trigger senescence, so fixing the structure directly fixes the leakage.
And our research team detailed a major finding here, right? The identification of a specific protein.
Yes, the protein KOX7RP. It acts as the structural glue that holds these supercomplexes together. It's the assembly foreman.
So what happens when you enhance the foreman?
When KOX7RP was overexpressed in mouse models, the animals lived longer, healthier lives. They were running a much more efficient cellular power grid. This upstream intervention reduced the damaging ROS right at the source, which is far more effective than trying to mop it up later.
That's an incredible breakthrough. And translating this to the clinic, we're seeing new ways to handle already damaged mitochondria through a process called mitophagy.
Yes. Mitophagy is the critical quality control mechanism, the selective recycling of damaged mitochondria. And our intelligence confirms there is now positive clinical safety data for small molecules designed to induce this process, like the candidate VNA318.
And how does a drug tell a cell to clean up its own power plant?
VNA318 works by creating a controlled, transient energetic stress. This subtle stress acts as a signal, forcing the cell to identify and tag the truly inefficient mitochondria for recycling. It's a huge step forward for neurodegenerative diseases, and VNA318 is one of the first of its kind to demonstrate safety in early human trials.
The lesson here is clear. The highest tech interventions are only as effective as the underlying efficiency of the body's most basic functions: sleep, metabolism, and energy production.
So if Section 1 was programming the immune system and Section 2 was stabilizing the hardware, this third pillar is all about turning biology into software. We are witnessing the industrialization of regenerative medicine.
And the current bottleneck for turning stem cells into usable therapies, like new neurons or tissues, is manufacturing. It's slow, it's expensive. You called it artisanal.
It absolutely is.
Really?
The technological solution highlighted by the intelligence we gathered is a platform from Syntax Bio dubbed Cellgorithm.
Cellgorithm. It sounds like an autonomous recipe, like genetic robotics. How does it automate turning a generic stem cell into a specific mature one?
It uses CRISPR-based logic, Geats genetic circuits embedded directly into the cell's DNA. It's like installing the instruction manual inside the cell and making it self-executable.
Okay, let's walk through that step by step. How does the cell read this internal manual?
So imagine the differentiation pathway is a three-step process, A, B, and C. For the cell to move from A to B, the genes for stage B have to activate at the exact moment stage A is complete.
Right.
The Cellgorithm system uses specialized guide RNAs that are initially blocked. When the cell successfully achieves the milestone of stage A, a Cas9 event triggered by the cell's internal state removes that block.
So the instructions for step B are locked until the cell signals it's done with step A.
Exactly. And that action instantly activates the guide RNA, which then turns on the genes for stage B. The successful completion of stage B then triggers the mechanism for stage C. It's a self-driving, robust cascade.
That's a truly elegant engineering solution. It cuts the timeline for producing these therapies from months down to days. This is the difference between a $1 million experiment and a scalable therapy for mass clinical use.
It's the democratization of cellular therapy, driven by synthetic biology.
Speaking of complexity, the sheer scale of aging biology is just too vast for humans alone. This is where AI excels. Let's talk about how AI is finding anti-aging compounds that researchers completely overlooked.
Right. So the problem is that a compound might look great in a petri dish, but fail in a complex system. The intelligence we reviewed highlighted a platform called Clockbase Agent. It analyzed over 2 million molecular profiles using not one or two, but 40 different validated aging clocks.
40 different clocks. That's moving beyond simple correlation into truly complex pattern recognition. What did the AI find?
It did two things. First, it confirmed the consensus compounds we already knew worked, like rapamycin and metformin, which validated its method. But the real breakthrough was its ability to flag overlooked compounds that had no traditional link to longevity science.
Give us an example. What did it flag?
The model flagged a compound called uabane, which is traditionally used as a cardiac glycoside. The researchers were so confident in the AI's prediction that they validated it in 20-month-old mice elderly animals.
And the results confirmed the AI's prediction.
They did. The elderly mice showed significant improvements across several domains: improved physical frailty, profound gains in cognitive ability, and clear restoration of heart function. It shows AI can connect disparate data points to find targets the human eye just misses.
So we have engineered cells and AI finding the molecules. The final piece of this programmable biology puzzle is the ultimate goal: partial reprogramming, turning back the epigenetic clock.
This is the field using modified Yamanaka factors to reset a cell's age. The goal is to achieve broad rejuvenation by resetting the epigenetic state, but without causing the cells to lose their identity or, critically, turn cancerous.
That balances everything. We want a youthful liver cell, not a tumor. How close are we to achieving that safe partial reset?
The field is rapidly advancing towards human translation. The research team noted the steady progress of candidates like ER100, which is on track to enter human trials soon for specific conditions, like optic neuropathies and chronic liver disease.
Why start with those specific conditions?
Because they represent clear, definable clinical endpoints where tissue repair is paramount. Preclinical success with ER100 has demonstrated enhanced tissue repair and reduced inflammation in these models. It's how you get regulatory momentum.
This whole shift from treating symptoms to treating aging itself by resetting the cellular instructions, it really feels like the core narrative tying these three pillars together.
It absolutely is. When you see reports of massive investment, with billions going into these multimodal approaches, the message is clear. Aging is now viewed not as an inevitability, but as a complex, engineerable systems failure.
The science is stunning, but the conversation can't just end with lab results. The critical next question is, how quickly and how equitably will these breakthroughs move from the lab to the patient?
That gap between discovery and delivery is vast. Let's start with a pipeline review. Where are these revolutionary therapies right now?
Yeah, let's get specific.
Okay, so the intelligence we gathered shows a steady progression. The fundamental understanding of aging, like DNA repair, that's still in basic research. But we have significant momentum in the preclinical and pre-IND stages.
So things that are getting close to clinical reality.
The CRT targeting UPAR for senescent cells is a great example of advanced preclinical work. The partial reprogramming candidate, ER100, is moving rapidly for ocular and liver diseases. But it's in phase 1 and 2 trials where we see the most immediate patient impact.
And what's in those trials?
Specific senolytics targeting BCL-XL, which we discussed, are actively being tested for diseases like idiopathic pulmonary fibrosis and chronic kidney disease.
Why those diseases in particular?
Because they have extremely high burdens of senescent cells in the affected organs. It makes them excellent measurable diseases to test the drug's efficacy and safety. We're also seeing stem cell therapies progressing through trials for frailty. The focus is still on treating specific diseases caused by aging. Even if the real goal is broader age reversal, it's the path of least regulatory resistance.
And across all these trials, the trend is overwhelmingly toward combination therapies, so no single magic pill.
That is the unequivocal conclusion. Aging is multifactorial, the solution must be multimodal. And a really provocative study we reviewed suggests that the sequencing of these interventions matters tremendously.
The order you do things in, why is sequence so important?
The idea is you have to clean the system before you try to boost its energy. For instance, clearing cellular garbage using things like rapamycin or senolytics should ideally come before metabolic boosting.
Like boosting NAD plus precursors, why would that be bad out of sequence?
Well, if you boost energy production with NAD+, in a cell that's already full of senescent debris and damaged mitochondria, you might just accelerate the production of more damage and ROS.
So you could actually accelerate aging, not reverse it.
Potentially. You have to systematically clean the house, remove the bad stuff, before you renovate and boost the power.
That brings us right to NAD plus optimization. We know levels decline by about 60% with age. The first idea was just to flood the system with precursors like NR or NMN.
Right, massive doses. But the new strategies are far more nuanced. Research now shows that the body's ability to use those precursors often becomes the bottleneck.
So what's the smarter strategy?
The new finding is to focus on supporting the enzymes that recycle NAD plus the salvage pathways. Using compounds like specific polyphenols to support these recycling enzymes may be more effective than just pushing in raw material.
So a stacking approach, combining precursors with recycling support, is superior for cognition and performance.
Precisely. It's about systemic optimization, not just jamming a massive input into a clogged system. We love hearing your thoughts.
Right.
Whether you're on YouTube or your favorite podcast app, leave a comment and let us know what you think about equitable access to longevity science. If you're listening on a podcast service like Apple Podcasts or Spotify, we'd be incredibly grateful if you could leave us a rating and review for Tomorrow Unveiled. It truly helps us reach new listeners.
The science is getting smarter, but the societal problem looms large: the cost and accessibility of these powerful therapies. This is a massive ethical and economic gap.
It's the most critical non-biological challenge. Our research compiled intense coverage on this. We have gene and cell therapies like CRE cells with price tags between $500,000 and $1 million per patient.
That cost structure immediately creates a two-tiered health system, a longevity elite.
It already exists. Longevity clinics are charging over 100,000 euros annually. This is what led Peter Diamandis to be quoted by the research team, emphasizing that healthspan must not become a privilege of the wealthy. The ethical implications are profound.
And it's not just the high cost, there's the safety risk of these powerful drugs leaking into the uncontrolled market.
Absolutely. We're seeing uncontrolled off-label use of promising drugs like rapamycin. The intelligence cited cases where healthy individuals taking it outside of monitored settings experience significant side effects.
What kind of side effects?
The clinical literature documents things like elevated blood glucose, increased infection susceptibility, and impaired wound healing. These are powerful pharmaceuticals, not advanced supplements. The risk is real without medical oversight.
So if manufacturing cost is the barrier, especially for cell therapies, what are the innovative economic solutions being proposed?
The financial shock of $1 million therapy is unsustainable. One key proposal is outcome-based pricing. The payment to the drug company is contingent on the therapy actually working over a defined period.
So you pay for the result, not just the product.
Exactly. Another proposal is amortization, treating the cure like a mortgage that's paid off over the years of healthy life you gain. And, of course, there are technological solutions, like creating off-the-shelf cellular therapies, which should drastically reduce the core manufacturing cost over the next decade.
This has been an incredibly detailed and expansive deep dive. Let's try to review the key takeaways from all this converging intelligence.
I think the biggest one is that the goal has decisively shifted from just chronological lifespan to achieving robust, active health span. The 100-year healthy life is now a viable scientific target.
And that achievement is being driven by this convergence. We're engineering immune cells to clear out debris. We're programming biology like software with things like Cellgorithm to automate regenerative medicine.
And crucially, all this high-tech revolution rests entirely on the foundation of low-tech fundamentals. The data shows insufficient sleep is a stronger predictor of mortality than diet or exercise, and that metabolic dysfunction dramatically accelerates neurodegeneration.
Right, you have to fix the foundation first.
You do.
And finally, AI is acting as our navigator, finding overlooked compounds like uabane that can accelerate our progress toward these sophisticated sequenced combination therapies.
Which it raises an important final question. The line between treating the diseases of aging and simply treating aging itself is rapidly dissolving. As these interventions move from targeting specific illnesses, like Alzheimer's, to reducing biological age across the board, what specific regulatory or ethical framework do we need to treat aging itself? The current medical system is designed to treat existing illness. A framework designed to promote universal health and reverse biological age that's still missing from the global regulatory landscape.
That is the ultimate challenge for the next generation of researchers, policymakers, and for all of us. The scientific machinery is assembled and working to make the 100-year healthy life a reality. Thank you for joining us for Tomorrow Unveiled.
We'll see you next time.